Okay, so thanks, Karen, for this great opportunity to chat to everybody. As you mentioned, I've chatted with you on and off, and I think there's some of my McGill colleagues on this call as well. So it's great to see there's a huge, rich, fragilized community across Canada. We kind of come into it in a slightly different way, I think, from some of the other people, especially my colleagues upstairs with Nahum and people like that who've come in from a sort of biochemistry perspective. We come into this really from a neurophysiology, kind of more of a standard neuroscience perspective. So I'm going to say something about my title in a minute because it's a little bit grandiose, and that's not something I usually do, but I thought I'd put that title because I wanted to emphasize something. So if anybody's familiar with the work that I do in my lab, then what we're really interested in is working out, identifying the building blocks of neuronal circuits and central synapses. And so if anybody's paid attention to some of the things we've done, we've looked at a number of different kinds of ion channels which are involved in that, both sodium channels, potassium channels. We haven't published a lot yet, but we've actually done a lot of work on it. We've also looked at inhibitory GABA receptors, so many of them are fundamental to neural circuits. And then, of course, if you know anything that we've done over the years, we spent a lot of time looking at inotropic glutamate receptors, so looking at glutamatergic synapses, and of course, they're fundamental for so many things in the central nervous system. Then you can study them and look at many, many different kind of angles. So as I said, I have this kind of grandiose title. You might think, well, why have such a grandiose title? In search of the origin of intelligence, which sounds like something out of a Carl Sagan a documentary or something like that. But what we've been learning, and I'm going to try and kind of take you through this, how our kind of reasoning of where we're getting to that. In a funny kind of way, we're kind of finding ourselves actually trying to look for the origin of intelligence. And we're kind of doing that by looking at the makeup of glutamatergic synapses. And so what I'm going to try and do over the next 30, 40 minutes or so is maybe just try and take you through how we've kind of reached that conclusion by looking at these building blocks of glutamate receptors. And then I guess in the context of Fragile X, but I'm really going to be talking about a broader sense by looking at intellectual disability in a broader sense, so not just telling you about the stuff we do in Fragile X, is that it looks like glutamatergic synapse is a really important hub in all these neurodevelopmental disorders. And we're sort of pinning a lot of things that we're finding there on that. But I guess the most important thing, and it kind of came up in the conversation with David Hampson and I were just having there a few minutes ago, is that we're finding that you have to understand new biology just about the existing, let's call it wild type behavior of neurons before you can go on and actually find insight into what's actually going on in these CNS disorders. So maybe the first person who really started to think about what is the origin of intelligence, and of course, intelligence is important to all of us because we care about intellectual disability. So that's where I'm really coming from it. But if you look at, if I was to take a walk across campus, and for many of you in the different universities where you are, then maybe I'll bring up my, maybe I'll show you my, do I have a laser pointing here? I can show you. Smart pen, highlighter, draw. This is a new system for me. Okay, maybe I'll just stay with this. Oops, all right. Can everybody see my mouse?
We can see it.
Yeah. Okay. So, if I was to walk across campus, if you go speak to some psychologists, then they would have a different definition of intelligence, perhaps, or a different kind of way to think about it than cognitive scientists. And then we've got a whole bunch of computational people here who might think about it a different way. But the way Donald Hebb looked at it, not necessarily head on, but the way he kind of thought about it, and it's kind of alluded to his book, and of course, many of you, because most of you are Canadian, will know that Donald Hebb was actually a Canadian neurophysiologist, and he worked within the buildings where I am. And he wrote this very influential book way back in 1949, right? So it's a long, long time ago. And he was looking at different kinds of species. And from these species, he concluded that the ability of man to have such a, complex and a deep thought, if you like, in terms of its intelligence was really tied into the sort of anatomy of the brain, right? And then as you actually work your way down the different species, the reason they have much simpler kind of behaviors is really tied into the limitations, if you like, of the brains. And I'm going to kind of come back to that, but more in a much more molecular sense as I get later on. So with that said, just go back to here. One thing that we all know, of course, is neurons are, the brain tissue is packed full of different kinds of nerve cells. And there's literally billions of different kinds of neurochemical synapses that's mediated. This is a snapshot through the hippocampus. And so there's billions of different kinds of chemical synapses there. And what I'm going to kind of emphasize to you, I think that the glutamatergic synapses is going to be really an important synapse to really understand in the context of these neurodevelopmental disorders. But to do that, I think we're going to actually have to understand new bits of biology about glutamatergic synapses, because what we've been teaching undergraduate students in the last 30 years or so, I think is really a superficial understanding of how these glutamatergic synapses work. So that's really what the rest of my talk is really going to be about. So just before that, just in the interest of just clarity for everybody and disclosure, I'm a co-founder and the chief scientific officer of this company called NOS Pharma, and NOS Pharma is actually trying to bring forward a therapeutic approach towards Fragile X syndrome. So just so that everybody's aware of that. I'm not going to talk about any of that work here, just for the interest of, because it's kind of part of that IP thing. So in terms of glutamatergic synapses, so if you're a university teacher, then you might show a slide, something just like this, to explain something about glutamatergic synapses. And so early microscopy using electron microscopy identified glutamatergic synapses, and they were asymmetrical in nature compared to inhibitory synapses, which were symmetrical. And so here's the presynaptic side. If you can see my mouse here, here's the postsynaptic side. And the thing that really struck all these early morphologists was that there was this electron dense region on the postsynaptic side that they called the postsynaptic density. And the reason it's electron dense, if I now move up to the schematic, is because it's packed full of different kinds of signaling proteins many of you will be aware of, and many of you have probably even studied them yourself. And so out of all of that cloning era that happened in the 90s or so, we're just chatting about Peter Seberg, Steve Heinemann's lab, Rick Hugener was part of that. I was kind of involved in a sense as a young kind of postdoc coming through the system. Many of these proteins were identified at that time. And so what happened at that time, we kind of got into the stereotypic view of how glutamatergic synapses work. And so there were two important ion channel families in glutamatergic synapses. The sort of champion or the king or queen of glutamatergic synapses was the NMD receptor. And that's because NMD receptors are highly calcium permeable. And they're very exotic because they've got this coincidence detection ability because they have this voltage-dependent block by magnesium ions. And so it's only when the cell gets excited, the magnesium block disappears and the NMD receptors bring in calcium. And that drives a whole bunch of learning and memory mechanisms that people have been studying for the last 30 years. The other ion channel, which is important there, other glutamatergic ion channel, which is important there, are the AMPA receptors. And you can see maybe this is a psychological thing in terms of the authors, the way they drew it, but they've kind of stuck the AMPA receptor on the outside. And that's because the way we really thought about AMPA receptors is that the AMPA receptors are really more of a peripheral, they have an important role because they're the first to get activated when glutamate's released, so they depolarize the membrane potential. But then the important stuff is all done by NMD receptors because magnesium block is relieved, and so they then get all this calcium coming in, and that's the really important thing. And so the AMPA receptors really just get things going. Now, there's a number of reasons why over the last 5 to 10 years, we think that's just probably not true. There's probably much more nuanced understanding glutamatergic synapses. And that's really because of two main reasons. So I forgot to mention, so there's been a renewed interest in this morphology. So the way we tended to think about it is that all these signaling proteins are all lumped together in synapses, but a lot of evidence from Tom Blompier and a bunch of groups out in Denver who have shown that actually within a synapse, there's actually specific nanodomains. And so it's designed to look like it house different kinds of signaling proteins in a spatially segregated way, even under the same presynaptic terminal. And then what I'm going to tell you about is how our understanding of these functional properties of glutamate receptors is expanding. But why did we think that the AMPA receptors might be doing much more than we really thought about? And so some of it was just based on just basic things that we were finding. So the first thing is that the AMPA receptor core subunits, which were identified way back in the 90s, by Seberg and Steve Heinemann, they identified the poor for some units, and then they just moved on. But then 10 years later or so, what we found is actually there's a whole bunch of different auxiliary subunits that actually bind around the AMPA receptor, and they have very profound effects on changing the properties of these AMPA receptors. And so that's something that my lab has kind of tried to champion in the last, 5 or 10 years. If you go and look at some of our papers, we've really tried to put into context how these different kind of families, how they're affecting AMPA receptors. So without diving into all the biochemistry, but a really important group is the claudin superfamily. There's 6 forms of these TARPs. I'm going to be telling you about them in a minute. And there's also this claudin protein, which is a bit of an oddball, but it turns out it's really important. It's called GSG1L. It seems to form this kind of unique role within the CNS. But there's also other families of auxiliary subunits. So there's cornichons, there's 2 of them. There's also these CCAMs or the shishas, depending on which part of Germany you come from. They seem to give them different names, but there's 4 of them. And there's also this other protein, there's a family of proteins called syndigs, which are really identified by Alva Diaz out in UC Davis, who found them. So AMPA receptors bind them in a whole bunch of combinations. But what's interesting now is we've started to build up a series of rules by which these AMPA receptors assemble. And so what it means is we can try to recapitulate what a native AMPA receptor looks like when you actually have, when you do it in a recombinant system. So something that's really striking, and so an area that we're interested in the cerebellum, is that most of the AMPA receptors in the cerebellum are all claudin-bound AMPA receptors. They either have TARPs on there, or they also have GSG1L, but they're all claudin. But the cornichons and the CCAMs don't seem to be found in the cerebellum. Whereas if you go to the regions of the brain, like the hippocampus, which is shown here, or in the cortex, for example, then not only do they have the TARPs bound, but they also have cornichons and these other ones. So I guess the important thing to get across, which is kind of summarized in these 4 or 5 rules that I've kind of come up with, which I'm kind of putting together in this big review, is the first thing is based on proteomic work and work we've done, is that most of the AMPA receptors contain this GLUE2 containing subunits. So that's not surprising. People were aware of that, but back in the 90s, so 30 years ago. But what's something that's fairly new is all of the AMPA receptors co-assemble with one of these TARPs. So there's usually at least two TARPs on the AMPA receptors. I'm going to come up to that in a minute. And the exception to that is GSG1L, and that's because GSG1L is a bit like a TARP. It's a claudin. It kind of tricks the cell into thinking it's a TARP. And so there's about 5% of all the AMPA receptors in the brain of GSG1L. I'm going to show you in a minute why that's kind of interesting, what GSG1L seems to be doing. Once you get into the cortex and the hippocampus, then not only do AMPA receptors assemble with TARPs, but they also assemble with cornichons. And remember, the cortex and the hippocampus are the brain regions which are involved in a lot of computational work. And so there's something about their presence that seems to be very important in that role. And you also get that combination of TARPs and CCAMP. But the important thing to remember is the AMPA receptors almost always assemble with TARPs. And when they do, they're binding with the TARP-like molecule called GSG1L. Okay, so that's the first reason why we started to think AMPA receptors must be doing much more than how people have been thinking about it in the past. The other reason, which kind of goes back into neurodevelopmental disorders, is that now, and this slide's already out of date, even though it's only a few months old, there's at least 100 missense mutations that have been identified on the pore-forming subunits of the AMPA receptors. So if you all remember, there's GLUE1, 2, 3, and 4. GLUE3 is particularly impressive in that almost all the half of the missense mutations that have been found on AMPA receptors are found in the GLUE3 subunit. If anybody's interested, we can chat about that at the end and why that might be the case. But what's interesting is if you look at the different portions of the protein, the amino-terminal, the ligand binding domain, and the transmembrane region, is that these missense mutations are found throughout the protein. And we were wondering, well, if AMPA receptors, all they're really doing is simply depolarizing cells so that magnesium block can be relieved and calcium coming in, then why is it so detrimental to have mutations all over the protein? It's not in specific areas. So how is that going on? And so the way we've kind of approached that is rather than just treating the whole protein as a, it's just one substance, we've sort of said, well, why don't we kind of reverse engineer the AMPA receptor? And so over the last 10 years or so, what we've done, and so here's some of the papers that we've published, we've identified a whole bunch of different kind of allosteric sites. So the ion binding site, which is in this dimer interphase, the claudin regulatory site. So this is the sites where the TARPs and GH2NL bind to the AMP receptors to regulate them. So we are the ones who identified them. They're actually in this area here. We also looked at alternate splicing through the flip-flop cassette. And it turns out the flip-flop cassette worked with the claudin sites. That's something that couldn't have been anticipated 30 years ago when Seberg and that were looking at things. So there's a lot of kind of interactions going on. And then recently working with a structural biologist, Teru Nakagawa, who's in Vanderbilt, and our lab did a lot of the functional work. We actually found that in terms of calcium transport of AMPA receptors, which people didn't think was very important, it turns out every single AMPA receptor's got this calcium binding site that sort of just sits outside a thing called the membrane electric field, which might not mean very much to everybody, but it's sort of a place where the ions start to send the voltage across the membrane. So it sits out there and it basically grabs calcium ions. I'm going to come back to that in a minute because it turns out this site G here is a very ancient, evolutionary ancient design of a protein. So almost all AMPA receptors had them. And then recently we just did a paper showing the QR site. So the site here is actually rather bendable. You can actually change its properties and so therefore change the property of the calcium permeability of AMPA receptors. So that's all basic science work. But the interesting thing, and I'm going to come back to in a minute, is that all of these residues, if you go back a slide, all of these residues that are found in the missense mutations in AMPA receptors, it turns out they all pile up in these areas that we've identified that seem to be important for the working behavior of these AMPA receptors. So we don't think that's a coincidence. We think it's because the moving parts of the proteins are the ones that are most susceptible to mutation. And so it gives us a roadmap to try to understand how to think about kids with these different types of mutations in these AMPA receptors. So I'm just going to give you a quick kind of run through of how that's changed how we think about AMPA receptors. So there's any electrophysiologists on the call, they will know if you record from different kinds of nerve cells. So here's the hippocampus and cerebellum, some examples. They all have different time course of their synaptic responses are all different. People don't pay attention to that anymore. But the reason that's important is because that is all determined by TARP binding to these receptors, right? So when different TARPs bind, you get slower or faster gating. And that also works with alternate splicing of the AMPA receptors. So TARPs are involved right at the very beginning in determining the time course of activation of glutamate receptors. The other thing has been known for a long, long time. So this is from Andrea Rossoff, but it's also Neil Bernashek was the senior author on this. He worked with Sackman a long time ago, and they noted that depending on the synaptic responses, you can get very fast recovery from inactivation. Desensitization of these receptors are very slow, but nobody could ever explain it just based on the different kinds of AMP receptor subunits. And so we've been able to explain it just only in the last few years. And it's because of this protein called GH2ML. It's a kind of TARP-like protein. It kind of pretends to the cell that it's a TARP, binds to AMP receptors, a bit like the TARPs. But what it does now is it causes a slowing of recovery from desensitization. So this behavior they see in layer 2-3 neurons, that are found in the cortex is actually due to GH2ML, something that couldn't have been anticipated before when people were cloning things because they hadn't identified all these auxiliary subunits. So it's really transforming how we think about AMPA receptors. So what it means is that the signaling properties of AMPA receptors are quite different. Now, what's interesting with GH2ML is you would have thought that that kind of turns up pretty early in brain development. But if you look at a rat and a mouse, so here's just some sort of images taken out of the paper we just had a couple of years ago, it turns out GH2ML turns up pretty late. So it turns out P60, a couple of months in of brain development. Usually you tend to see all these proteins turning up in the critical period, P14, P21. But GH2ML turns up late, and because GH2ML slows up the signaling capacity of AMPA receptors, what we're finding, we haven't published yet, but all the other TARPs actually express much earlier. And so they don't slow up recovery from desensitized much. And so what you have is this slowing down in the sort of excitability of the cortex as it develops, which is kind of interesting. And it kind of explains why maybe, for example, kids with Fragile X, that maybe the expression pattern of some of these auxiliary subunits is disrupted, and that might explain the hyperexcitability in these tissues, and something we can chat about at the end if anybody's interested. So that's how these auxiliary subunits are changing how we think about the fidelity of synapses, or how synapses signal within glutamatergic synapses. But what about calcium permeability? So for the last 30 years, ever since they were cloned, there was kind of two types of AMPA receptors. There was the GluA2 containing AMPA receptors. So here's my little schematic. So the GluA2, the GluA2 subunit is this kind of gray here. And the reason they thought they were calcium impermeable is because the GluA2 subunit in the narrowest region of the ion channel pore had an arginine residue there that was placed there by RNA editing. And arginine is positively charged, and so the thought is it repels calcium ions that are trying to go through the narrow pore region of AMPA receptors. And so consistent with that, if you do some recordings from Purkinje cells, so this is in the cerebellum, and so it's been done by Peter Jonas and a bunch of other people 30 years ago. This is data from our lab that we did just to repeat it. And what you find is the way to measure the calcium permeability is you look at their responses in solutions which are rich in sodium ions. You can see you get this very nice linear relationship, which is what you expect because you get high sodium on the inside, high sodium on the outside. And if you remember, painful as it might be, if you remember your Nernst equation, then the reverse potential is going to be close to 0 millivolts. However, if you replace all the external calcium from the outside with high levels of calcium, then you can see the reverse potential goes very, very negatively. And that tells you that calcium is not very good at transporting across these cells. So these are not very calcium permeable. In contrast, in the same region of the cerebellum, there's these non-neuronal cells called glial cells, Bergman glia, very famous. They've been studied a lot by many, many people. What you can see is when you look at the responses in high sodium concentrations, high levels of sodium, you get this, what's called the biorectification of the IV. And that's because of this thing that I identified 30 years ago, can you believe it? With Mark Mayer when I was at NIH and a student, this thing called polyamine block. And the occurrence of this polyamine block has been tied in to the occurrence of calcium permeability of AMPA receptors. And just consistent with that, if you replace all the external sodium with calcium, you can see you get a positive shift in the Versa potential of these AMPA receptors. And so you get this high calcium permeability. So this is standard things that we teach to undergraduate students, and I've been teaching this for more than 20 years. But there were some reasons for us to think that this doesn't make sense because these are AMPA receptors that would never exist in the brain, because in the brain, they're always bound by these auxiliary subunits. And so we wondered what would happen if you try to recapitulate something that was much, much more likely to happen, to exist in the CNS. And so what we did is we turned to the cerebellum again because we'd recently published a paper where we'd worked out the stoichiometry of different AMPA receptors, ones which are in the stellate cells. So these are interneurons, which are found in the sort of distal part of the molecular layer of the cerebellum, and also the Purkinje cells, which I just told you about before, which aren't very calcium permeable. And these are these big, beautiful Purkinje cells, which you find here, the Purkinje cell layer. And so what we did is we took knowledge of their stoichiometry in terms of the TARPs, recapitulated them, and asked, does the presence of TARPs somehow change their calcium permeability? And it turns out, surprising to us and of course to everybody else, is it does actually change their calcium permeability. So this is what was done 30 years ago. Well, we did it, of course, again, but we recapitulated what everybody's seen. Here's the sodium response, but here's the calcium response. You see, it's very negative. So it's telling you there's not a lot of calcium permeability there. When we added 4 TARPs to the AMPA receptor, so we were able to tether them, and we could talk about it later, but we know that recapitulates the data channel. You can see you're getting a sort of movement more and more close to 0 millivolts reversal potential, suggesting there's some calcium permeability. Same thing when you have 2 TARPs, but on the GluA1 subunit of these AMPA receptors. But what was remarkable is when the TARPs are on the GluA2 subunit, and so remember, the default thing is when you form heteromers in the brain of AMPA receptors, the TARPs first go on to the GluA2 subunit. When you do that, you actually get a very remarkable shift in the reversal potentials. Now, less than minus 10 millivolts. So that makes them actually pretty calcium permeable. So here's just a summary of that. So this is how people used to think about it 30 years ago. What we found is you actually start to move the calcium permeability up. So it's almost sodium's about 2 times better than calcium getting across the membrane. But that's impressive, right? Because remember, there's a sea of sodium ions outside cells, about 150 millimolar, and there's only a few millimolar calcium. So somehow this channel has been able to come up with a way, working through this site G, to capture the calcium ions and ignore the sodium ions. So it's now starting to transport calcium. Okay. Now, of course, in the cortex and the hippocampus, then, which is all the kind of computational parts of the brain, then they co-assemble not just with TARPs, but they co-assemble with cornitions. So here's a structure, a pinch from an old friend of mine, Ingo Gregor, who's the LMB in Cambridge. And he had the structure, it was published a couple of years ago in Nature, where he showed the TARPs are bound in the BD positions, just as everybody's seen before. But the cornitions are actually in the AC position. So they bind to GluA1, whereas the TARPs begin to GluA2. And you can see the structure is a little bit different. Cornitions have got a slightly different structure from the TARPs. So we wondered if we co-assemble an AMPA receptor heteromers with TARPs and with cornitions, does that give you even more of a boost? And that would make sense, but if you go into brain regions like the cortex, hippocampus, etc., that extra calcium would really change things from a computational capacity. And that's exactly what we found. So here's the 2 types of cornitions. There's a cornition 2, there's cornition 3. And when we co-express with cornitions, you can see you get this big shift. It's now almost close to 0 millivolts. So you're now getting almost as a strong ability to transport calcium and sodium. And we did it with cornition 3. It was even bigger effect. It actually went beyond. It went up to plus 10 or something like that, millivolts. So that's a huge change in calcium permeability of these AMPA receptors. Now, what's really interesting, if you remember, I told you about the Purkinje cells, the Bergman glial cells, they have this rectification due to polyamine block. But you can see there's absolutely no polyamine block here. So that kind of signature that has been used for the last 30 years to identify calcium permeable AMPA receptors no longer holds true, because it would look that they're not polyamine sensitive, but they're actually calcium permeable. And this calcium permeability, I'm just showing you the summary, it's as good at transporting calcium as sodium. So that would have a real impact on activating different kinases and phosphatases, etc., and glutamatergic synapses. So that kind of simple idea of what we had, where NMD receptors kind of dominate everything, and all the calcium permeability comes from them. But calcium's coming through these AMPA receptors, and they're obviously regulating things too. Something that we don't fully understand yet, but it's something we're interested in. So that's a new biology, and it kind of broke the mold a little bit of how we think about things. So when I go back in the fall now, I'm going to have to teach things. I'm going to have to skip me work. I'm going to have to rework my lectures to teach the undergraduate students. Now, yeah, I got it wrong for a little bit, but here's how we're starting to think about it. Now, let me try and bring it back now to kids with intellectual disability, because that's the thing that we really want to understand. But to get to this point, we had to understand new biology. And then when you put that new biology in, and you start to look at some of the kids with intellectual disabilities. So this might be a bit too technical for everybody, but here's a kind of sequence alignment of the different kinds of glutamate receptors you can find. The cytan-LAF, that's this region here that we identified for a calcium binding site. So it's highly conserved amongst all glutamate receptors, so NMD receptors as well. So this is a calcium binding site that nobody was aware of before, that we just identified a couple of years ago. Here's the QR site that Peter Seberg and Steve Heinemann identified 30 years ago. But now we realize there's an actual interaction between them. That's the first thing. But the other thing is, we actually go into the clinical literature. We found there was a couple of kids who had missense mutations at the QR site, and also at this other site called the +4 site. And so we were interested to see how that changed the calcium permeability to these receptors. So if you look at all the receptors together, you see, rather than forming just this two types of calcium permeability, no calcium permeability, high calcium permeability, then you actually get this continuum of calcium permeability of AMPA receptors. So you can go in and choose whatever calcium permeability you want for an AMPA receptor. So that's a bit of a game changer in how we think about things. But what about these kids with these missense mutations? So one of the missense mutations is here. And so what you see is that suddenly now they're very, very high level of calcium permeability, because the A1, A2 would be a way down here, right? But now it's suddenly get this high level of calcium permeability. If they then associate with the TARP, which is what would happen in the CNS, they've got an even bigger level of calcium permeability. Remember, this is a logarithmic scale, so this is a huge difference, right? And likewise with the arginine to the glutamate, which is a negative charge, so it's even more calcium permeability to see. So what we found is that we could kind of draw a dotted line across looking at calcium permeability. So if the calcium permeability of AMPA receptors is about, it's as good as transporting calcium as it is sodium, then for whatever reason, that seems to be fine in the CNS. But it's once you get into being twofold, threefold, or fourfold, even better, then for whatever reasons that we don't fully understand yet, then that dysregulated calcium transport seems to be giving rise to at least autism and particularly intellectual disability, at least in these kids here, right? So one can go off and do all of these. I'm going to come back to in a minute how we think this is maybe a general principle that we might be able to use to understand some of the missense mutations that people are finding. But somebody might say in the audience who's been around a long time, David Hampson and I were chatting about this, how come nobody's ever seen this range of calcium permeability before? And it turns out they did. It's just that people ignored it. So here's data that came from Peter Joris's lab. So in this paper, he had Neuron literally 30 years ago. With Jörg Geiger as the first author, he's now got his own lab. You can see they found whole ranges of calcium permeability. We've also gone in and repeated that. We get the same results. We also measured the polyamine blob. And so they did have this observation. They didn't have no calcium permeability, high calcium permeability. They actually had a range of it. And if you go into that original paper, when they did it, the reason they misunderstood or kind of interpreted things incorrectly was two main reasons. First of all, Peter, and this is not really a criticism on his part, is he assumed, because everybody at the time assumed that AMPA receptors were pentamers. And then the biggest assumption that turned out not to be correct is they assumed that GluA2 subunits could actually have a variable stoichiometry on AMPA receptors. But now we know it's fixed at 2, so you can't have this variability by changing the number of GluA2 subunits per AMPA receptor, because they're always fixed. So it means that this must be due to changes in the auxiliary subunits. So the data was there, right? And so why is that important for everybody here who's interested in intellectual disability and glutamate receptors? And the reason it's important is if you now look at the different sites that we've identified, so here's a structure that we have of the AMPA receptor when it's transporting calcium and water molecules through the pore. So you can see it's quite a big kind of highway of transporting of ions. But then if you take all the missense mutations that we found from other people's work that have been reported in the literature but haven't been tested functionally, what you see is that this pore region is absolutely packed with missense mutations. Look at the one for GluA2. GluA2 is, of course, extremely important because that's the one that's got the arginine residue here. You see there's already four amino acids actually in this QR site area that are actually mutated and cause very, very severe intellectual disability in these kids. But they're also found in the Cytan site. So what we think is that many of these mutations, which are shown on this slide, will have the same properties that we found for the 2 or 3 mutations that we looked at more carefully. So if you're teaching undergraduate students and you still haven't really bought the idea that AMPA receptors, most AMPA receptors are actually quite calcium permeable, then if you know anything about your evolution, then you shouldn't be surprised by that, because if you go back in time, so here's the sequences of these regions. So let's go back a second. So here's the QR site here. Here's Cytan LAF, but here's now the amino acid sequence of the QR site here. And here's the Cytan LAF. You can see, as other people have found before, there's a glutamine, GLUA1, 3, and 4, but no GLUA2 because it's edited to an arginine. And that's what people thought caused most AMPA receptors to be calcium impermeable. That's not true anymore. Here's the Cytan LAF. That's the site G site, this site, which is up here, which sits outside the membrane electric field. And if you go back into like aplysia, which is this kind of sea slug, a lot of people have studied historically in terms of neuroscience, you can see that they also have a cytosol, so they can bind calcium, catch it like a kind of basketball. And they also have a glutamine there, right? At these sites, even the KIT receptors. There's an arginine in the glutamine, but it turns out that's not because of RNA editing. It's because of some other reason. It's just part of the sequence, right? So it turns out in aplysia, there is no evidence that there's GLUE2, that there's calcium permeable AMP receptors. They're all calcium permeable. And then if you go down into a trichoplax, and I'm going to show you what a trichoplax, if you don't know what it is, in a minute, what a trichoplax is. So this is a very simple multicellular organism. I think it's the most simplest but multicellular organism it's known, at least to science right now. You can see that all their AMP receptors, they're called AKDFs. So these are the evolutionary ancestors of AMP receptors that diverged from the path that mammalian AMP receptors took about 600-700 million years ago. You can see that they all have a glutamine in their pore region here. So they were calcium permeable. There was never a calcium impermeability of AMP receptors. You can see the cytosol site is a little bit different, but there's a lot of the elements in there that means that it's very similar to the mammalian receptors. So what that means is that these proteins, these AMPA receptors, maybe even for the AMD receptors, but for the AMPA receptors for sure, which is kind of surprising because we've kind of ignored them for a long time, is that they were calcium permeable for the longest of time. And it was only during, as we got into the vertebrate brain and RNA editing somehow was discovered by biology, that we toned down that calcium permeable AMPA receptors. And I'm going to get to that just in a minute, but just to remind you of, if anybody's unfamiliar with the trichoplax. So we've got a paper coming out where we've looked at the properties of these receptors, the trichoplax. So this is how trichoplax eat. So they're kind of moving around. They're kind of in the sea. They're only about a millimeter or so across. They can move around. And that movement here is actually driven by these ancient AMPA receptors. And it turns out when they consume, the different kind of algae, etc., do it again so you can see it, when consume these different algae, then it opens up these glutamate receptors, calcium comes in, and that's actually the thing that's actually causing this movement here. So if you take a blocker of AMPA receptors that would shut down a human brain and put it in a dish with these trichoplax, then that activity is completely eliminated. So what that means is that the proteins that we now need for us to have this high intelligence and our ability to reason and understand our world really started off as a way for organisms to actually feed, right? It was just a way in which to move around and find foodstuffs, right? So the AMPA receptors and all the glutamate proteins, which eventually get co-opted into this synaptic structure, then they've been around for ages, right? And it's only recently, in evolutionary terms, that they've been co-opted in to form these kind of synapses, right? So what makes me say that? So the reason I'm saying that is if you go back, and so this is more of a schematic, and look through the different kind of species by evolution, all the way down to trichoplax and even to organisms even simpler, what you see is that the glutamate receptor subunits have been around a long, long time. They go way back 700, 800 million years, right? Because that's when these organisms branched away from the mammalian line. But the things that we hold dear to in the context of mammalian AMPA receptors, which is important for the function of our brains, this RNA editing, which we were talking about for the QR site, then that really only turned up with vertebrates, okay? Really only turned up then. And likewise with alternate splicing, which we think is very important for switching on and off the different types of TARP regulation, then that also turned out pretty late, okay, in brain development. Likewise, if you look at the axillary subunits, same thing. They really only turned up in vertebrates, and they weren't around much before that. Now, if you look carefully, you can see the cornitions, they go back. They're very ancient proteins. But it turns out they weren't associated with AMPA receptors at that time. They're more involved in cell cargo and transport of proteins in that sense. And there's some evidence that maybe TARPs were around in, for example, C. elegans. There's some evidence for that, but if you want to look at the data where people have published on that, I'm not sure if I'm completely convinced that that's the case in Drosophila. I know there's these other proteins called BETOs, which are found to the KIT receptors there, but it seems something that was needed when vertebrates were evolving and you made this rather complicated brain. It's only then you started to bring in all these extra levels of regulation and that the AMPA receptors started to associate with these auxiliary subunits. Before that, glutamatergic synapses, when they became synapses, so here's the nervous systems kicking in here, then they would have been devoid of all of these. And that might be one of the reasons, one of the biochemical markers explaining why their ability to kind of master the world that they're living in is limited compared to what a human could do. So how does this fit up into other things? So in terms of if you're looking back over the last 30 years, then the work that was done in the 90s, all this cloning work that many of us kind of came out of being grad students and were involved in the cloning of different glutamate receptors, in this case, the AMPA receptor subunits, the discovery of alternate splicing by Peter Seberg and Heiberg. He also discovered RNA editing, the actual biological biochemical mechanism of RNA editing. He discovered it by looking at AMPA receptors. So these were done 30 years ago, and they really represent the first chapter of the glutamate receptor, the glutamatergic synapse story. But what's happened is that we've kind of taken that, and most of the field has kind of moved on and assumed that nothing has really changed since. And so if you open up any textbook, that's kind of all the thing they'll ever talk about. But what's happened in the last 10, 15 years or so is that we've identified much, much more about the assembly of AMPA receptors. So it's an assembly process that happens within the ER. And so the TARPs are added there. There's some debate about whether or not the cornitions are added, but the TARPs, the claudin-type proteins, are added there. So they're the default for all AMPA receptors. And as you've kind of shown, they have profound effect on not only the gating of AMPA receptors, but the permeation. And it's still not really fully understood yet, but there's these other proteins, the shishas and the C-caps and the synapses, etc. They turn up before they get put on the surface. So it's not really understood how that works. So why is this important? So one of the reasons it's clearly important, it comes out of our GSG1L paper, is there's some synapses that seem to be TARP-dominated, AMPA receptor synapses, and GSG1L. So one of the roles of these auxiliary subunits, not only are they changing the functional, the gating, the permeation properties of AMPA receptors, but they're also changing telling the cell where they have to be put. So it's setting up different signaling channels within the same cell. And you might say to yourself, well, if that's really true, then shouldn't you expect to see a very complex biochemistry within the synapses of all these auxiliary subunits? And that's exactly right. So now if you go and look at the work's been done by a number of different kind of groups. So there's Tom Sudoff, who of course did all the neurexins and neuroligand stuff. There's work from Anne-Marie Craig and Tabri Siddiqui, who's been looking at RTM dependent mechanisms through neurexins. And there's also groups in Japan who have been looking at LGA1 and McGill and ADAM22. So there's a whole host of different ways in which AMPA receptors, glutamate receptors, come together to form these synapses. And so it makes sense that the AMPA receptor, the different kinds of AMPA receptors that you find would be very diverse in their functionality because it ties in to very, very diverse functionality, which is found in all these proteins that basically form the scaffolds to set up these synapses. I'm almost finished, so just give me one more minute or so. What's, of course, been well known from Tom Sudoff's work and other people is that many of these proteins that are listed here, missense mutations in them, not surprisingly, they cause intellectual disability and autism in kids. So it's all channeling all to the same place. The genetics of it are different proteins are involved, but they're all going to the same kind of marketplace. And I think that's why we kind of feel that the glutamatergic synapse is a real hub for this. Maybe not the only one, but it's a really important hub for these neurodevelopmental disorders. And that's something that I haven't really talked about, but that's something that we've really embraced when we've gone after things and trying to understand things in the context of Fragile X syndrome. So just to finish, I just want to thank the two people who are involved in the Nature paper. Two first authors, there's Fedi and Shintong. There's also Edward Yan, who had finished up his master's and moved on, but they were all the two first authors. I want to thank the funding we have. And then all the other work we've done, I just want to thank everybody who's been in the lab, who were partly involved in this. And of course, the collaborators who have been fantastic in helping us solve these problems. And of course, thank you for your attention. I didn't intend to speak as long as this, but I hope it hasn't been too boring, and I'm very happy to ask any question, answer any questions, and chat about things. So thanks.